So I would just like to share some prospects of new vaccines targeting COVID-19. Right, so these are the outline of my content for today's presentation. So I will touch on a few aspects on why do we need to vaccinate and various platforms that we have for as vaccine candidates for COVID-19, and what are the candidates in phase 3 clinical trials at the moment. Right, so vaccines are actually the cornerstone of fight against communicable diseases. So it has been proven that vaccines are the most economical or effective strategy to actually prevent and eliminate infectious diseases. This has been proven by the success of smallpox eradication in the 1970s, the drastic reduction in polio cases during the past 20 years, the progress towards tetanus elimination, and also the reduction of measles mortality. Despite these achievements, infectious diseases are still responsible for nearly 30% of all deaths worldwide. This is more than 15 million people dying every year, right? Mostly in low and middle income countries. Till date, I actually was checking on the Corona tracker this morning, and over this morning to just now, there were about 300 more deaths, all right, just in a few hours. Okay, so total till date, there has been about 846,785 deaths worldwide due to this COVID-19 infection. So how close are we to COVID-19 vaccine? So the US FDA only approved the first vaccine against Ebola virus last year, 43 years after the deadly virus was discovered, all right? And you know that we had this Ebola in maybe end of December 2013 up to about June 2016, right? So on average, it takes about 10 years to actually develop a vaccine. With COVID-19 crisis looming, everyone is hoping that this time it will be different. Vaccine makers are racing to develop COVID-19 vaccines and have already advanced a few candidates into clinical trials, but we still have very challenging times ahead. So this is taken from GSK, a pharmaceutical company. So the industry average on the vaccine development cycle, actually, this cycle actually mentions that from laboratory identification of antigens up to about to market the vaccine or use the vaccine for patients, it takes about 10 years roughly. So why do we vaccinate? Right? So in the first diagram over here, you can see that if you don't vaccinate, all right, and you're infected with this virus, you actually tend to fall ill from it. So your body's response naturally is to fight against it, right? And then it will tend to produce these antibodies. So it depends also on age. It probably depends on other comorbidities, and it depends on several factors or your immune system, your body's immune system itself, on how effective it is to actually fight this virus. All right. So eventually, when you recover from it, your body will produce these antibodies, right, and confer immunity towards future infections. But with vaccine, all right, in the second case, you do not fall ill, right, as in the first case over there, right? So it sort of like mimics the entire an infection process in the body, all right, without you actually falling very ill from this vaccine. And your body will still, your immune system will still be provoked to produce these antibodies against these viral antigens, all right, and it will still confer the immunity towards the virus. All right, so this is the two cases on why do we need to actually vaccine, all right, comparing when you don't vaccinate. So looking at this, all right, to actually identify a vaccine candidate, first you would need to actually know the viral structure, or in microbiology we call this as morphology. So suitable candidate can be identified by knowing the functional aspects of these antigens. So all these antigens that we have for COVID, SARS-CoV-2 virus has been described by our previous colleagues, right, who presented on this structure of the vaccine. And there are several candidates here. There are, or several proteins on the surface of this virus. They are the spike proteins, and then inside you have the nucleocapsid protein, and then you also have envelope proteins, and you have membrane proteins, all right? So since S proteins and its fragments are prime target for developing SARS and MERS vaccines, all right, so it is also expected that similar regions of the SARS-CoV-2 could also be considered as clinical targets for COVID-19 vaccine, all right? And also, since genetic sequence was already released in January 2020, right, just this year, more than 40 pharmaceutical companies and academic institutions from many countries have actually engaged in developing vaccines for COVID-19. So over here is just a diagram showing you there are several vaccine candidates that can actually elicit an immune response, right, in the host cell. So vaccine candidates can comprise of the whole virus, or it can be from the nucleic acid, or they can make vaccines through the viral vectors, or it can be protein based as well, right? Okay, so currently there are about 8 technologies of platforms that are being tested for COVID. In the classical platform, this were actually used many years in developing vaccine prior to the new generation platforms that we have today. All right, so traditional vaccine development use these classical platforms, which uses the whole inactivated virus. They use live attenuated virus, protein subunits from the virus, and also viral-like particles. All right, and then we also have next generation platform. So this platform uses viral vectors. They use DNA or RNA, and they also can use antigen presenting cells. All right, so these are the 8 different platforms that we have for testing or to develop vaccines. So the first one we look at is the live attenuated virus vaccine. This contain live virus particles that have been weakened so that they do not cause a disease, all right? They are cheap to produce. They generally give good immunity against infection, but they can also induce cell-mediated and antibody-mediated immune responses. Concern of this vaccine is that it could back mutate and also become virulent, right? And it's unsuitable to use in immunocompromised individuals where they have suppressed immune system. There are 2 attenuated candidates in preclinical development at the moment using this technology. The second one here is known as inactivated or kill virus vaccine. Okay, this contain whole virus particles as well, but without the genetic material, right? So the RNA and the DNA, or the DNA of the virus, is usually destroyed chemically or by heat, leaving the immunogenic elements unaltered. Okay, so generally it is safer than live vaccines, and it can actually confer immunity, but at a lower rate, they said, all right, compared to the other vaccine, the live attenuated vaccine. So this is generally more stable also, okay, without refrigeration, and it is considered very popular in the low and middle income countries. Next one is subunit vaccine, right? So this is made from some antigenic proteins of the virus without any of the genetic material. That means they cannot replicate inside the body, and they are very safe to use, right? So the immune response is induced by this subunit vaccine is not as strong as some other types of vaccines. So in this case, they actually add, or they use it with adjuvants. Adjuvants are some chemicals that can bind to this virus to elicit a better immune response in the body. So several doses may be given to be as effective as the other vaccine candidates. And currently, there are about 36 subunit vaccine candidates for SARS-CoV-2 in development. Right. The next one here is known as the viral-like or virus-like particle. Right. Known in short, we call it as VLP. They mimic real viruses, but contain no genetic material. All right. So the protein shell is synthetic, and they mimic the actual virus. Okay, but they don't have any genetic material inside. So it can actually induce strong immune response. It's not infectious, and it is known to be more stable than some subunit vaccines at extreme conditions. But they are more difficult to develop, right? Than some other vaccines, like subunit vaccines are much easier to develop. They may also require multiple doses to be effective. And there are 7 candidates of this vaccine that are currently being developed for COVID-19. Then we also have the use of viral vectors. Okay, so in viral vectors, we can use 2 types. One is replicating viral vectors, which means that we can use viruses like measles virus. All right, we can just use the measles virus and insert some genes of SARS-CoV-2 antigens into the measles virus genome as well, and then allow them to actually replicate in the host cell, right? So when it replicates, it actually produces the antigens of the SARS-CoV-2 as well. So this can be detected by the immune cells and also confer immunity this way, right? So one potential disadvantage is that prior immunity to the vector may render the ineffectiveness of this vaccine, meaning, like, let's say, for example, in this diagram, they show that measles virus is used as the vector, and when a person has MMR vaccination, right, then it would produce antibodies against measles virus. So in this case, when you give this virus, so the antibodies may actually attack these vectors and actually destroy them, all right, before it can even reproduce or elicit any immune response against SARS-CoV-2 antigens, right? So that is what it means. However, there are some of these this replicating viral vector that are being tested currently. And then we also have non-replicating viral vector vaccines. This non-replicating viral vector vaccines typically use adenovirus that have additional genetic alterations, meaning they have removed the capability or the genes that are capable of replicating in the host cell, right? So these viruses, these vectors, when you insert the SARS-CoV-2 genes in them, it would not actually replicate in the body, all right, but exhibit the antigens of the SARS-CoV-2, and then it can elicit the immune response, right? And then there are some vaccine candidates in clinical trials at this moment using this non-replicating viral vector. Next is a nucleic acid vaccine, all right? So we can use DNA from another bacteria. So this DNA is in the form of plasmid. And plus, in this plasmid, you actually insert the gene that encodes the SARS-CoV-2 antigen, right? So when it enters into the host cell, it actually can reproduce and produce the mRNA of that SARS-CoV-2, and then it produces the antigens, right? And then these antigens can be detected by the immune cells and confer immunity. So such vaccines have several potential advantages over the conventional types. In this includes the low production cost and also storage, right? And but the thing is, because this antigenic proteins is produced inside the body cell, a stronger immune cell-mediated immune response is induced, as well as the antibody immune response. And there are several candidates that use this technology. And then we also have nucleic acid with RNA. So when you use RNA from SARS-CoV-2 virus, it is actually enclosed in a lipid delivery vehicle, right? So this liposome, we call this as a liposome as well. So this can actually carry the mRNA of SARS-CoV-2 into the host cell, right? And then because it has got the ready mRNA, then it will be just translated into producing the antigens of the virus, right? And then this can be recognized by the immune cells to confer the immunity. However, this is less, because RNA is less stable than DNA. So far, historically, there has not been any vaccines, right, that has mRNA, okay, as a component of the vaccine. So, but then, however, there are some one of two candidates using this RNA technology, and they will be in clinical trials. So, okay. So these are some of the vaccine candidates in phase 3 clinical trials, right? So I've just listed all those that I've got from the WHO document that have been registered. Okay, so we have from AstraZeneca and University of Oxford using the non-replicating viral vector using adenovirus as the vector. Okay, we have another one, Sinovac, using inactivated virus vaccine. Wuhan Institute of Biological Products with Sinopharm also uses inactivated viral vaccine. And then we also have Beijing Institute of Biological Products also using inactivated vaccine. Moderna, National Institutes of Health, right, from the US, using RNA technology. BioNTech with Fosun Pharma and Pfizer is using RNA technology. So these are the six uh, companies or developers that have got certain of these vaccine candidates already that are registered for phase 3 clinical trials. And these are some of number of projects at various stages using various technologies that I've mentioned before. Okay, so I think by now this information I obtained that was published in May 2020, but I suppose by now there are more industries or more developers. Okay, so far, right, from what I got from the WHO document, there are 6 vaccines that have entered phase 3 clinical trials, and there are 142 COVID-19 vaccine candidates that are undertaking preclinical evaluations. There are 31 COVID vaccines candidate that have entered clinical trials, including phase 1, phase 2, and phase 3. And these are just comparison, right, from what I got for SARS. Okay, so SARS Technology, they have used these 2 platforms actually to develop their vaccine, DNA and also inactivated virus. And this is for MERS. MERS uses modified vaccinia virus vaccine, right? This is a pox virus as a viral vector. DNA, they also use adenovirus as a viral vector. So the efficacy evaluation is also required, right? So a sponsor of the vaccine would actually need to provide data from placebo-controlled trials indicating their vaccine is at least 50% effective against COVID-19, according to the FDA guidelines, all right? This is effective 30th June. Safety of the vaccine is also very important, right? So it is very important as what I've highlighted here. The AIDS Foundation had actually called on, right, global public health leaders or community and governments to actually to exercise caution, right, and due diligence in fast tracking the vaccine candidates for prevention. So they have also highlighted the fact not to cut corners on safety and efficacy, because it's foremost, it is 2 very important aspects in actually implementing vaccine for use in our population. So there are these number of challenging situations ahead of us. Most vaccine candidates that make it to the preclinical testing hardly even make it to the market. This is by statistics that most of the time, 90%, 94% fail, all right, to make it to the market, all right, even from clinical trials. So we have a chance of 6% that will actually make it, all right. So in this case, that is why we need many developers, all right, many sponsors, many institutions to take part in developing multiple vaccines using various platforms, all right, so that may actually have a better chance in getting some vaccine that actually works in the future. different vaccines have different pros and cons, right? So some may require more doses than others. So manufacturing, testing, distribution will also vary in this case. Some vaccines may be more suitable in some populations than others, right? So this can be due to age, geographical region, genetics, all right, other risk of diseases that different patients would probably be undergoing. Clinical trials would also need to be carried out in these various age groups, right, and different geographical regions. So does any country have the infrastructure, resources, and planning capabilities to meet the demand of COVID-19 immunization while still meeting current vaccine needs? So planning and implementation of policies, infrastructure, and funds are greatly required. So I guess you know that developing vaccine is actually $1 billion investment, all right? And if it fails, then all these $1 billion will actually be really wasted, all right? So proper planning, policies, infrastructure is also needed. And ways to maintain the cold chain, all right? Cold chain is very important to maintain the properties of the vaccine, especially when vaccines are actually to be transported to rural communities, all right, where transportation and electricity, all right, could actually hinder or break the cold chain, right, in maintaining the properties of this vaccine. So ways to maintain the cold chain is also required. Public fear of vaccine. So a lot of people are actually frightened or actually fearing of even going out of their house to to contract or get a virus, all right? Even when they sneeze, they are so afraid that they might have COVID infection. So public actually fear of vaccine. So what if someone is called to be a subject for these clinical trials, and you actually have to tell them the disadvantage or the adverse effects that might happen to them. So already they are fearing of getting COVID-19 infections. But if you are, if they know that they may actually end up with some kind of detrimental effects on them, then it can actually delay subsequent trials and also delay launches of these vaccines. So, I would like to share a quote by Stephen Barr. He's an associate professor of microbiology and immunology at Western University in London, who said that the best vaccine in the end may not be the best for everyone, but the second one might be for those who don't respond, right? So it's always good to have these backup vaccines as well, or vaccines that can be used in parallel around the world. So these are the new additions and updates that I got from this month, all right, in the month of August. So we have Merck, Themis, and Institute of Pasteur, all right, vaccines will enter phase 1. CanSino Biologic announces phase 3 trials in Saudi Arabia. Russia approves a Gamaleya vaccine before phase 3 testing. Okay, so this is going to be very controversial for many people. Vaccine given through the nose could also protect against infection. This is a very interesting news that I got, but then they have actually tested this in mice, all right, not yet in humans. Italy's Rethera goes into human trial of COVID-19 vaccine. Johnson & Johnson will begin the large late stage of 60,000 person involved in their trial in September. Okay, so before I end my talk, I will just leave you with a short presentation that I have. I'm not sure if it's going to open, all right? Okay, I'm so sorry. I think it's not.
Okay, Dr. Priya, I think thank you very much for your excellent presentation.